All right, thank you, and let's get started. So thank you for joining us today for another round in our webinar series on investigations of biomedical issues and how they relate to human disease. First, I'd like to thank Gary Watkins, Director of Marketing at AnaBios, for putting together this webinar series. And as a quick intro for those who have not joined us in the past, can you go to the next slide, please? Yeah, so AnaBios is a unique contract research organization and a biotech based in San Diego. We recover human organs from ethically consented donors and use tissues or cells to conduct physiological assays in which we test preclinical compounds for drug discovery projects. We've essentially redefined first in human. And working with AnaBios is as close as you can get to a clinical trial without actually doing a clinical trial. For those who are developing therapeutics for respiratory disease or renal disease, AnaBios provides precision cut lung slices and precision cut kidney slices from both human lung and human kidney. Lung and kidneys are recovered from postmortem donors with both normal condition and disease pathology. These tissue slices retain the 3D microenvironment of the tissue and have become the go-to tissue model for validating therapeutics for fibrotic diseases, and provide the data to advance therapeutics to the clinical stage. Slices can be shipped to clients for their in-house R&D. However, in addition, AnaBios provides services measuring inflammatory and fibrotic markers in precision cut lung slice, such as you see here. AnaBios uses both in vitro fibrotic models, such as the TGF-beta treated tissue slices, as well as tissue slices from diseases such as IPF and CKD. These services can advance your therapeutic program by providing human relevant data on the efficacy of your compounds. If your team is interested, our scientists can design custom studies relevant to your program. So now returning to our webinar series with the next slide, I'd like to highlight our next pulmonary focus webinar. The next webinar in the pulmonary series will be Dr. Mehdi Maneshi from AbbVie. Dr. Maneshi will present on the use of human lung translational models to advance pulmonary fibrosis research. Now, final technical points regarding the webinar. We'd like to hold the questions until the end of the webinar. To submit the questions at that point, please use the chat box. You should see a chat box on your panel on the right where you can type in questions, and I will read them out to today's presenter, Dr. Salzman. So with no further ado, it's my great pleasure to introduce today's speaker. Dr. David Salzman is CEO and scientific co-founder of the Gatehouse Bio. He earned his PhD in molecular biology from the University of Connecticut and completed his postdoctoral training at the Yale School of Medicine. His academic work focused on the role of microRNAs in precision medicine, uncovering mechanisms for microRNA biogenesis and activity, and the development of widely used research tools such as Cy-Check2 dual luciferase reporter assay and the Dicer monoclonal antibodies. He continued working on precision medicines at Biogen and Miradex, leading to PKD and BD bioanalytics to support IND applications and clinical trials. Notably, he played a role in advancing Qalsody, a FDA-approved therapy for SOD-ALS, and PROS-Tox and PREV-IO-Tox, commercial diagnostics that predicts the tox to radiation and immuno-oncology therapies in cancer patients. At Gatehouse Bio, Dr. Salzman oversees the company's R&D pipeline and serves as the scientific visionary behind its mission to develop oligonucleotide-based precision medicines for complex diseases. In 2024, Gatehouse Bio was awarded the Translational Research grant from AnaBios, and today we're pleased to welcome Dr. Salzman as he presents a deep dive into microRNA-driven mechanisms of pulmonary fibrosis and the research that led to GHB1589, a first-in-class therapy designed to treat specific patients with pulmonary fibrosis. Dr. Salzman, the floor is yours.
Great. Before I begin, I'd like to thank Gary and Richard for the invitation to come and present, as well as for all of the attendees who are taking the time out of their day to come listen to the talk. So we're seeing a global shift towards precision medicines. We know over the last 20 years through clinical trial results that precision medicines are resulting in better patient outcomes, as well as greater likelihoods of success for FDA approvals. In addition, this is causing a shift towards more value-based pricing models as opposed to cost-based pricing models for these drugs, which makes them highly lucrative for the pharmaceutical industry. Precision medicines today, though, are limited. They're targeted against around 3% of the genome, looking specifically at protein mutations, and they're focused largely in the oncology space or rare genetic disease space, which have their own challenges due to things like labor-intensive screening or small clinical trial populations that hinder speed. We believe that precision medicines of the future are going to require new druggable targets outside of the protein coding genome, mainly in the transcriptomic region, and that utilizing this area of the genome for biomarkers and drug targets will enable different types of AI modeling that can mine data for targets and also design therapies prior to the first major investment decision that needs to be made in the R&D process. However, there's a question, which is where will the next precision medicines come from? At Gatehouse Bio, we believe that small RNAs provide a unique opportunity for developing new precision medicines, enabling research into the 80% of the transcribed genome. At Gatehouse Bio, we focus on small non-coding RNAs, which are non-protein coding transcripts less than 200 nucleotides that encode for genes like microRNAs, siRNAs, piRNAs, endogenous and other types of endogenous small non-coding RNAs. The discovery of microRNAs in 1993 really unlocked the whole reservoir of druggable targets and therapeutic opportunities, ultimately winning the Nobel Prize in 2024 after becoming really one of the fastest growing areas in biomarker and therapeutic research. At Gatehouse Bio, as I mentioned, we're focused on all of the small non-coding RNAs that are less than 200 nucleotides, and you'll see they all form kind of these classical hairpin structures or duplex structures. This is very important in the biogenesis process pathway, where each small RNA species or class is processed by various types of RNase and RNase-3 enzymes to generate these short 18 to 24 nucleotide small non-coding RNA, which is guided to a complementary target site in a messenger RNA, thereby inhibiting its translation into protein. Over time, what we've learned is that small RNAs are master regulators of gene expression that control key biological processes and pathways, and they have been implicated in pretty much every single disease and indication that's been studied to date. As I mentioned, they are one of the fastest growing areas of biomarker research, with over 1,500 clinical trials being executed testing their clinical utility as either biomarkers or therapeutics since the year 2000. Now, a lot of work has been done on microRNAs, and I want to kind of lay out some of the previous work that's really laid the foundation for what we're doing at Gatehouse. Some fantastic work coming out of Ian McRae's lab back in 2012 showed a high resolution crystal structure of a small RNA co-crystallized with the Argonaute protein, which is the catalytic engine of the complex known as the RNA-induced silencing complex, or RISC. And what we know from long string of biochemical work, genetics, X-ray crystallography, is that the microRNAs or small RNAs that are loaded into the RISC complex have functional domains, and they're positionally located within these genes. The 5' terminal nucleotide is very important for defining AGO loading. We know that nucleotides 2 through 8, referred to as the canonical seed region, are very important for messenger RNA targeting. We know that complementarity between regions 9 through 12 have to do with the mechanism of action, signaling the RISC complex for targeted messenger RNA cleavage or translational repression if they're not bound. And we know that there are other compensatory binding sites in important regions in the microRNA or small RNA that occur between position 16 and onward. Now at Gatehouse Bio, we built a very large relational database, a structure-activity relationship, after testing the interaction of over 2,500 microRNA mRNA combinations, doing various mutations in positional location, site-directed mutagenesis to build up this database, enabling to have enhanced target prediction capabilities. However, there is a limitation in the field, and one of the limitations is that the current databases that are used to annotate microRNA genes contain about 2,600 human microRNAs. These genes were used to do target prediction using target prediction algorithms like TargetScan and others, which have a roughly about a 50% accuracy or 40% accuracy. So there's a little bit of room for improvement there. In addition to that, we know that there are a lot of vendors who have created kits and off-the-shelf reagents for either shotgun cloning or the specific detection of these microRNA genes, which work very well. However, they're missing a very large pool of the information that is available to us for biomarker research and therapeutic drug development. We know that these small RNAs, when processed or found in samples, are actually represented as arrays of sequences and not just a single gene. These arrays of sequences are called isomiRs or isoforms, and they occur through the canonical biogenesis of these small RNA genes, resulting in two types of isoforms. We can have templated isoforms, where we see additions or deletions from the canonical loci or mapped region in the genome. These are formed through alternative processing of Drosha or Dicer. And we can also find non-templated isomiRs, which are, they contain nucleotides at either the 5' or 3' end that do not map back to the genome. And these nucleotide additions are added by enzymes called template-independent terminal nucleotidyl transferases. And what they do is they change the sequence. And in changing the sequence, they change the biology of the small RNA itself, as well as all of the downstream qPCR or probe reagents that would be used to detect these genes at scale. Because these small RNAs are expressed in every tissue and every system and have application everywhere, we knew that we would want to be able to scale our discovery as well as scale the biological modeling of these genes. So we started to establish a platform that can do this work. So here's kind of what's going on under the hood a little bit. So we know that these small RNAs can be expressed as these arrays of sequences, these different isomiRs, which you can visualize on gels. However, current approaches actually at the molecular level condense this information, but they also condense the information at the computational or read mapping and quantification level, bringing these genes down to really a single positional location, which we can lay out the coordinates if we try to map out the 5' and 3' end sites for where where the coordinates would lay on the genome. By decompressing this data at Gatehouse Bio, we see these very large structures or information structures, which we can use for not only discovery, but also for like machine vision purposes. As we start to do this at scale, we find many different types of isomiRs and isoforms that are being expressed in various samples. The easiest ones to describe are these qualitative differences, where we may find the absence or presence of particular small RNAs in either healthy or diseased subjects. And we also find quantitative differences, where a particular isomiR may have low expression in disease or high expression in healthy, or vice versa. Using our QSAR models, we are able to predict out the biology and targets of these isomiRs, linking their expression and targeting to particular messenger RNAs and target sites, fitting those back to pathways, and then ultimately fitting those back to particular diseases that we can then start to interrogate for therapeutic purposes. And in doing so, we believe that we've unlocked the potential for small RNAs as precision medicines. The current gaps, again, just to reiterate, they are that isomiRs are not really well characterized and that no tools exist to model their biology, much less use them to stratify patients or design targeted therapies against them. And our solution to this is a system or platform that we've developed called CodeBreaker, because we believe that we're breaking the small RNA code. There are 4 major components or modules to our platform. The first one is simply characterization and featurization of all the small RNA content and isomiRs in a sample. And in doing so, we've been able to establish a proprietary database of information that really fuels our research, as well as different types of disease maps and biology maps. We then run analysis to try and discover what we would believe to be the druggable small RNA ohm. These would be genes that are differentially expressed or binary expressors only found in certain patient populations. We run various types of biological modeling to see if the small RNAs can stratify patients into novel endotypes or precision populations, where we can use the small RNA as a biomarker to identify those patients. And then ultimately, we use programmed drug design to develop targeted oligonucleotide therapies against those small RNAs that are dysregulated. And the oligotherapies that we develop can either be similar to an siRNA, which would be like a mimetic, and we would use those in the instances where the small RNA is downregulated or has loss of expression, or we'd use an antisense oligonucleotide to block the overexpression or aberrant expression of any small RNA found to drive disease. Over the last six-ish years, we've really been on a mission to generate as much data and map as much of the small RNA ohm as possible. To date, we have over 30,000 patient samples that we've sequenced and have processed and analyzed using our CodeBreaker platform. Here's kind of like a broad list of the data that we have. It's very hard to describe a lot of the data because the EMR data is a combination of cross-sectional and longitudinal samples with electronic medical records. There's lots of demographic and disease-related information. Some of it is specific to the either pathology or idiosyncrasies of each disease that we're studying. And ultimately, what we're seeking is precision drug targets. And today, what I'm going to be talking about is the work that we did that led up to the discovery and development of GHB1589, which is our lead precision medicine for the treatment of pulmonary fibrosis. So the discovery of GHB1589 really starts with the analysis of over 3,100 patient samples spanning 27 independent studies, of which Gatehouse Bio has contributed 55% proprietary process data from all of our partnerships. The data span 12 different matrices, 10 data acquisition platforms, and as I mentioned, represents a variety of cross-sectional and longitudinal both biological samples and metadata that we can fit into patient continuums to do different types of analyses to build associations. Highlighted in blue here are the studies from Gatehouse, and everything else is open source data that we're able to ingest and structure and recharacterize in our system, opening the aperture onto all of the isoforms that were found in the data. The results of our work identified what we called a hotspot that mapped to a loci in chromosome 13. This hotspot was defined because there were a lot of isoforms coming from it. There were about at least 25 isoforms per sample, per matrix, per disease, per patient. All of the isoforms were very high expression. They had high frequencies in the data set. And more importantly, they had uniform dysregulation in all of the samples that trended in the same direction. This is a very unique attribute to this particular discovery that was really critical for the development of this program. So we conducted a meta-analysis across all of the data sets and calculating the Hedges G, which is really like a test for trend, where we can look across multiple different studies as a way to kind of normalize all of the data and find trends. What we noticed was that these chromosome 13 isomiRs were pretty much universally downregulated. So the way this works is anytime you would see a negative value here, that implies that the or it indicates that the chromosome 13 isomiRs in aggregate were downregulated in that disease set. What we found was that these isoforms were reduced in at least 7 different diseases. And I say at least because this is really what we just were able to analyze to date, and obviously in the future, as more studies come into our database and we're allowed, and we have the opportunity to analyze them, the list of indications may grow. P-values can always get, enhanced. Things can become more significant, or, things can change. Things may not be significant as we add more studies, and there's always reasons for that. One thing that we found very interesting here was as we looked at the pulmonary indications, which is this big block of data over here, we had very strong signal in indicating that very, very high levels of downregulation of these chromosome 13 isomiRs were linked to disease. One of these studies over here actually comes from laser dissected fibrotic foci comparing the expression of microRNA isomiRs in the fibrotic foci to normal adjacent tissue. And that kind of very strong signal seen in the data set kind of really pins not only like the cell types, but also the importance of the signal that we were finding in the tissue of interest. Another thing that we found that was very interesting was, as you can see, we didn't find downregulation in every single indication. And the 2 indications that we studied here, one of them being COPD, showed that the signal that we were finding was really caused by or enriched in restrictive pulmonary diseases and not obstructive pulmonary diseases. So even though there is a strong fibrotic component in all of these indications, we would surmise that, or we would hypothesize that the fibrotic signal that we're finding in the COPD patients was not necessarily caused by loss of expression of what might be driving disease in IPF patients or other types of interstitial lung diseases. Finally, another big component of the research is that we found that the signal translated across tissue and a variety of biofluids, giving potential for longitudinal and disease surrogate biomarkers, as well as patient selection biomarkers as we go into the clinic to try and identify which patients may be applicable for therapy. Having the all of this data available to us allows us to do additional tests to build up the level of association and correlations to different types of clinical endpoints, so that we can weight different targets based on their disease association. So at the company, we never come up with a single target. There's always big panels of targets that we have, and we have to interrogate them to try and put essentially weighted averages on them to determine what we're going to wind up screening in the future. One of the tests that we were able to run with these particular data sets had to do with mapping the survival probability with and without these isomiRs. So in IPF, we're fortunate enough that there is a standard gap index model, which is used to predict the risk for mortality over 3 years. And what we found was that, including these small RNA isomiRs, we were able to increase the 3-year mortality by almost two times, and we also identified 2.5 times more high-risk patients by identifying subjects who fell into these kind of intermediate or low-risk groups and sorting them correctly into this high-risk group, which you see here in red. Looking under the hood, when we went to go see which features were important in the models, we found that the chromosome 13 mapped isomiRs were some of the strongest, tightest correlated, having the highest hazard ratios and lowest p-values again, indicating their association with disease and their importance in disease. And even more importantly, what we found was that a lot of the features that we identified performed even better than the standard GAP features, which is, sex, age, and physiology. So it's very interesting that the molecular biomarkers can actually perform better than the physiological markers. Using data that was generated from human lung biopsies and mouse lung biopsies, we showed that the signal translated. So we've translated now between human and mouse, which would be a critical system for doing drug development and further research. What we found in both cases was that either associated with disease, severity or GAP score, or time from bleomycin induction, was that these chromosome 13 isomiRs were all downregulated, and that there was an inverse relation between the expression of these isomiRs and the putative targets that we had, that our system had predicted that they regulate, which all fall within the fibrotic pathways, pro-fibrotic pathways, things like COL1A1, integrins, and fibronectins and TGF-beta receptors. So moving into data that Gatehouse generated, where we are now seeking to validate all of the initial findings that came off of the Codebreaker platform. We acquired a large bank of patient primary fibroblasts, and using these fibroblasts, we were able to do small RNA sequencing, and we also characterized the COL1A1 protein expression. And just as we found in the previous slide, we found a very strong correlation, both in pairwise or along a continuous scale, where low expression of these chromosome 13 isomiRs equated to very high expression of COL1 protein and vice versa. We also developed a targeted RT-qPCR assay against a handful of the isomiRs, and we correlated their delta CT values to RNU44. And what we found in screening, almost over 350 blood samples, these are now packed gene blood RNA from patients with various pulmonary fibrotic indications, various interstitial lung diseases and connective tissue disorders. We found that these chromosome 13 small RNAs were also downregulated significantly in these patient samples, as well as in fibroblasts or stellite cells collected from either IPF patients or patients diagnosed with non-alcoholic steatohepatitis. Because we wanted to test a downregulation model, we were able to acquire and generate animals that have heterozygote knockout in the chromosome 13 loci from which these small RNAs are being expressed. And we induced pulmonary fibrosis using the bleomycin model and observed that the heterozygote animals with haploinsufficiency had faster progressing disease, where we were measuring the change in body weight over time. They lost more body weight compared to wild type animals. They also lost it at a much faster rate, and they also never recovered where the wild type animals did. In this study, about 30% of the heterozygote knockout animals were euthanized due to significant loss of body weight, modeling in a way what we found in the human population, where loss of expression of these chromosome 13 small RNAs associates with low survival rates or high mortality. And finally, blinded staining for collagen on the lung samples showed that heterozygote knockout animals had significantly higher collagen levels compared to wild type, thereby validating, again, the findings from the Codebreaker platform. So knowing that we have all this data and that there seems to be strong impetus in IPF, we decided to use that as our lead indication, or our beachhead, to start doing drug development against. IPF is an orphan disease with high unmet clinical needs. It's there's only 2 FDA-approved therapies, which do not really improve the symptoms and have a lot of problems with tolerability. Areas where we see that oligonucleotides can really differentiate themselves from small molecules. And we would be able to have an advantage there. In addition to differentiating from, orally dosed small molecules, we know that we have a precision biomarker, which we can use to identify fast progressing patients. And the idea is that, upon patient intake at a pulmonologist, we can do a blood sample and test for the, isomiR biomarkers. And if the patients have low expression, they would be included in, or they would be candidates or included in the drug trial. If they had high expression, they would be excluded. This allows us to enroll treatment naive subjects as well as early stage, disease subjects, and potentially setting up any potential therapies to be frontline, because they can be, patients can be tested for and selected and identified at the earliest, point of intervention in the tertiary clinics. The biomarker data itself would enable a 52-week clinical trial with fewer than 200 patients, where we have a lot of ability to augment kind of like survival curves as well as FVC ratios and other types of clinical endpoints like the number of hospitalizations that a patient has over a year, as well as other type of biomarker endpoints. So this really set up the research strategy for us to go develop the precision oligo, whereby we would start synthesizing various small RNA isomiRs that came out of our initial discovery research and test them using multi-system testing in cells and precision cut lung slices and animals to understand the pharmacodynamics, kinetics, the safety, delivery, and as well as competitor analysis to see if we were actually performing better than standards of care and clinical competitors. And alongside this, we would also develop the biomarker assay for patient selection. This would be used ultimately as a companion diagnostic come clinical trials, where we would use that to select patients to give us a very homogeneous population, thereby increasing our probability of success or likelihood for getting clinical endpoints when treated with the therapy that we're developing. So as I mentioned, we selected a number of chromosome 13 isomiRs that came out of our data set. We selected 22, highest priority. They went into hit to lead screening and lead optimization. During this time, we were doing a lot of in vitro screening, where we used a tool delivery vehicle for oligo delivery. And we have actually since then selected a LNP nanoparticle formulation that we will be using going forward into GLP toxicology and clinical development. And I'm going to show a little bit of the data that we've generated on that fully established compound called GHB1589 using in vitro, in vivo, and ex vivo systems. The therapeutic design for these compounds is almost analogous to an siRNA. It's a double-stranded 20 to 24 base pair duplex. The antisense strand would be the active strand of the API. The unique thing about our drugs is that the antisense strand is derived directly from patients. And then obviously we do a lot of lead optimization to do chemical modifications to enhance its stability. As I mentioned, we've selected a lipid nanoparticle. This particle has a retinoic acid ligand that selectively targets STRA6 positive cells in the lung tissue using an intravenous route of administration. And the MOA, once taken up into the cells, is that the mimetic will be unwound and loaded into the RISC complex, where it will then target all of the requisite target messenger RNAs to slow, stop, or reverse fibrosis. One of the reasons that we selected the particular LNP that we did is because these STRA6 positive cells had very high levels of enrichment for all of the target genes that we had predicted these chromosome 13 isomiRs to be targeting. So it seemed kind of like a very serendipitous kind of hand in glove strategy for drug delivery. So knowing that there was a path forward, as I mentioned, we synthesized at a small scale 22 different chromosome 13 isomiRs They were, purified. They passed all of their, QA as well as QC metrics, where we test things like purity and baseline activity using a reporter assay. And all compounds being roughly equal, we decided to go screen them now using a bank of 9 different patient primary fibroblasts that we have acquired through various partners over time. These fibroblasts were characterized for both COL1A1 and isomiR expression. And in doing so, we were able to really stratify them into 3 groups here, which again models out what we're finding in the human population and brings us closer to clinical trials. We started calling this almost like in vitro clinical trials, where we're able to see if we can get good effectiveness in kind of really this first group of fibroblasts here, as well as kind of the second one, and see kind of what the differences in effects would be. The activity screening that we did was done on using 2 clinically or biologically relevant assays. The first one was looking at inhibition of collagen, and the other one was looking at inhibition of fibroblast cell growth. What we're looking at is a heat map showing the relative percent inhibition in each assay. It's the average of 4 technical replicates by 2 biological replicates. Looking at these on kind of a 2-component, on like a XY scatter plot, what we were ultimately looking for was compounds that had, or isomiRs that had the strongest activity in both assays. So any compound that would be down here in this bottom left-hand quadrant, where we saw inhibition of cell growth and inhibition of collagen, would obviously move its way into the lead. And here, as you can see from the data, that there was one particular isomiR that stood out from the rest of the pack. This isomiR was called mimic44, and we also identified 5 additional backup compounds that were also used in our preclinical research. However, I'm not going to be showing all of that data here today. Zooming in on the M44, you can actually see here it's very specific, reduction in collagen using quantitative ELISA or qualitative immunofluorescence, as well as its inhibition of cell growth using IncuCyte to monitor, cell growth or confluency on the plate over time. Using this assay also allowed us to measure the morphological, any types of morphological changes that we may see. And it was very interesting that mimic44 did not have any, aberrant or visible morphological or toxicologic effects on the cells that were being grown. And what it truly did was it kind of stopped them dead in their tracks from growing, which is a good thing. So we're not really trying to kill cells here. We're trying to stop fibroblast cell growth. Next, we wanted to compare the activity of mimic44 to other standards of care or clinical competitors. And here, what we're showing again is the activity of each compound described, labeled in the plot here, using 2 different assays, again, for inhibition of cell growth or reduction of collagen. And what we're plotting here is the EC50 value from a 4-parameter fit derived from an 8-point dose response curve with each compound. As you can see from the data, GHB1589 outperformed the next nearest competitor by at least a factor of 10, indicating that we are, GHB1589 has a lot more potency than other compounds that are being developed. And that led us to, ask questions around, well, how could that be? Why would that happen? As we start to look at the, fibrotic pathways and, profibrotic networks, it is a tangled mess of protein interactions and signaling pathways. There are at least 6 primary pathways involved, and that is not, really even getting down to the, very specific tissue level. where there may be, other components involved or other, signaling factors involved. But we know that integrin and FAC, TGF-beta, Wnt and beta-catenin, connective tissue growth factor, interleukin signaling, as well as positive feedback through collagen and extracellular matrix signaling play a major role in the profibrotic pathway. And because there's so much involvement, a lot of the drugs that are being developed today are very selective inhibitors of a particular component of the pathway here. GHB1589 provides a very differentiated approach where, again, we identified this particular chromosome 13 isomer, which we're naming ISO1589 as, the lead isomer with the most activity in controlling this pathway. It's a conserved gene with reduced expression in IPF patients. And as you can see, it targets multiple genes that sit across many different aspects of the profibrotic pathway. And, this type of targeting has been evolved over hundreds of millions of years by nature to, make this happen. And what we're doing now with GHB1589 is we're using a chemically modified oligonucleotide mimetic that restores the ISO1589 function in patients that have reduced expression of this gene. All of that prediction was verified by treating patient primary fibroblasts with the M44 sequence and then testing the targeting using RNA sequencing. And what we found was that, a lot of the genes that were being targeted and downregulated were within the Wnt, TGF-beta, and integrin alpha regulatory axis, and pathway enrichment showed, significant downregulation in pretty much all of those primary pathways, as well as pathways involved in cell growth, which makes a lot of sense because we actually saw that manifest through one of our biological endpoints that we were using to screen the drugs in the first place. So a lot of overlap in the mechanism and biology. Next, we wanted to test if GHB1589 formulated in a lipid nanoparticle could rescue bleomycin-induced pulmonary fibrosis. What we showed is that treatment of mice who have undergone the bleomycin pulmonary fibrosis model were essentially rescued using GHB1589. Treatment of GHB1589 rescued both inflammation as well as lung fibrosis and hydroxyproline, which is a measure for interstitial or pathogenic collagen deposition as induced by the model. Using fully formulated GHB1589, we went to retest now in the patient primary fibroblasts, where we showed that, using this final formulation, we recapitulated reduction in collagen, as well as knockdown of several biomarkers in the mechanism that we've defined now. Next, we moved to the precision cut lung slices, which we received from Anabios. And using those precision cut lung slices, we were able to test two different endpoints, looking both at the lung tissue itself, which we can see here, where treatment of the lung tissue with GHB1589 resulted in significant reduction of biomarkers in the profibrotic pathway at the messenger RNA level, and also in secreted biomarkers in the media, where we showed that, again, treatment of these precision cut lung slices with GHB1589 reduced secreted biomarkers in the media, which can also be used now to set the translational strategy for GLP toxicology in clinical trials, giving us good biomarker endpoints to test early drug efficacy in patients. Finally, using a Cy5 labeled GHB1589 compound, we treated the precision cut lung slices and did a lot of histology to understand what cell types the drug was being targeted into. And we saw uptake in both healthy tissue as well as disease tissue coming from IPF patients. The drug was being taken up selectively into type 2 alveolar cells as well as macrophages in both healthy and disease, in addition to aberrant basaloid cells and other cell types in the IPF patients. I should note that the durability of the compound was seen for at least 96 hours, which is as long as we were able to run the experiment for. However, the durability of oligos is well understood to be upwards of 30, 60, or 90 days, and we anticipate seeing that level of durability as well when we go into systems that permit us to test PK/PD out that far. Finally, we know that the GHB1589 therapy is able to penetrate deep into these lung slices, which is very interesting to us because you can imagine these lung slices as almost like a disc sitting on the plate, and as we put the drug on top of it, it has to sink down, and there's a question of, will it just stick to the top of the slice or will it actually penetrate through? Here you can see on the right-hand side the cross-sectional, this is a reconstructing of the cross-sectional images taken on the microscope. And you can see here in purple or fuchsia how deep the oligo actually penetrated into the lung tissue. Skip that. And finally, to understand the full scope or broad application of the M44 or M474 compound that we are testing, we tested its anti-fibrotic activity in primary cells taken from patients representing heart, lung, liver, joint, eye, skin, and kidney. What we found was that in treating diseased cell lines, we saw a significant reduction of collagen, as well as reduction of all the disease-associated biomarkers that are in our panel at both the mRNA level and qualitative immunofluorescence. And it also prevented TGF-beta induced collagen expression in all the different cell lines as well, including knockdown of the profibrotic panel that we're using. So again, verifying that GHB1589, or the target that we discovered in all of the different diseases, using our meta-analysis, is actually a conserved mechanism for controlling fibrosis, and that the gene that we discovered, or the target that we discovered, is truly a pleiotropic effector that has been kind of conserved or designed by nature to play a critical role in preserving this type of biology and keeping us healthy. Finally, this sets up a kind of portfolio play for us for the M474 or GHB1589, where we can test multiple indications right away using the formulation that we have now. Our goal is to get the drug development up through a stage of IND readiness. And we're also evaluating other types of delivery vehicles that would enable therapeutic accessibility in things like diabetic retinopathy, chronic kidney disease, or rheumatoid arthritis. And finally, it takes a village to do all of the work. I'm very grateful to all of the dedicated researchers and scientists who have helped us, along the way, including our team of highly dedicated researchers who span both, biological science and data science, as well as all of our advisors, both clinical and research. We would not be able to do all of this work without you, as well as partners at Charles River Lab and the Beth Israel Deaconess Medical Center. And finally, a special thank you to the IPF patients that we work with directly at the company. We see you, we hear you, and we're working towards, developing a cure for you and your families. So thank you very much.
Thank you, Dave. Thank you for a very comprehensive webinar. And so now we've come to the Q&A portion of the webinar. I did want to note, I didn't note it earlier, for those who wanted to get some information on the PCLS at NMBIOS, there is a handout section, so there's a brochure that you can download. So let's start with the questions. As I mentioned earlier, there is a questions portion where you can type in your questions and I'll read them out. Though actually, if the first question actually is going to be oral from Dr. Getty, CEO of NMBIOS. So Gary, if you could unmute Andrea, then you can ask the question and I'll follow up then with the other questions from the audience. I would recommend, Andrea, that you type it in via the chat box since we can't hear you, and they will just proceed with the questions. Dave, again, a very comprehensive webinar. I'll ask the first question and then I'll proceed to the others. Regarding the meaning of that hotspot, the chromosome 13 hotspot, you see, these isomiRs that are dysregulated in this section. Is there any correlation with some of these target genes that you also identified as being upregulated? Is there a meaning to this particular location?
Yes, absolutely. So, the Codebreaker platform in and of itself is a multi-omics, approach to, target discovery. However, we like everything that we do at the company starts with small RNAs. So our mission is to identify small RNA biomarkers first, and then see if the rest of the biology fits to the hypothesis. So in this particular case, we also have a lot of messenger RNA sequencing data, and the mRNA-seq data that we were analyzing after identifying the hotspot all kind of pointed in the right direction, indicating, targeting of the profibrotic genes and the profibrotic pathway.
Okay. Well, thank you. That's very interesting. So here's one question from one of the attendees. Have you considered delivering these oligos locally to the lung using like nebulation to enhance specificity delivery?
Yeah, so we did a large technology assessment to look at routes of administration. We believe, based on where oligo LNP delivery is in the world today, that the intravenous route of administration would be better suited as opposed to inhaled. So we did try inhaled and we did not have a lot of luck. So.
Okay, thank you. Another question is whether or not you've tested the outcomes that you're reporting with the in vitro assays are influenced by inflammatory cytokines, which may then affect the outcome. So in other words, is the crosstalk in the immune system, is that an important assay?
That's a good question. So we know a lot of the GHB1589 targets are also overexpressed in macrophages and other inflammatory cells. I think what we saw using the in vivo systems is that treatment or exposure to GHB1589 reduced inflammation, reduced the amount of infiltrating leukocytes and other types of fibrotic markers. So it would appear that GHB1589 has both it's kind of like an anti-inflammatory and anti-fibrotic effect, although the majority of genes, when mapped back into pathways, link back to more fibrotic than inflammatory pathways, if you will.
Right. I suppose one way to address this question is you could actually even use the PCLS as a way, as the in vitro model, where you could retain some of this crosstalk. The next questions are the oligos reactogenic? Do they induce RIGI or TLR pathways? And the second follow-up on that is, did you actually see any of these liquid nanoparticles in the liver?
So based on RNA-seq data, using the patient primary fibroblasts or lung tissue from the bleomycin animals, we didn't see any targeting or upregulation or stimulation of the toll-like receptor pathways.
Mm-hmm.
The second question is, did we see, I guess essentially, did we see any drugs sinking to the liver?
Right.
The easy way to answer that is yes, but a very small amount. The majority of the compound sinks to the lung tissue.
Okay, thank you. So the next question, so there's a lot of interest in combo therapies. So have you done a comparison where you looked at the GHB compound compared to the standard care, both alone and isolation as well as in combination?
Yes, I'm glad somebody asked that. So the answer is yes. We have tried every type of combination under the sun using the patient primary fibroblasts. We've done pre-treatment with standards of care followed by GHB1589. We've done a GHB1589 first, then standards of care, treat them at the same time, titrate one, titrate the other. We really don't see any synergy between the two. Interestingly, when we look at the patient data that we have almost 1,500 IPF patient samples in our database, and we can actually, we have all their EMR data. We don't see any linkage between treatment with perfididone, nintedanib, AZA, or any other therapy, inclusive of O2 replacement, that appears to impact or modulate the expression of these chromosome 13 isomiRs. They appear to be truly independent of that.
Mm-hmm. So would that lead to additive, or are you seeing additive, or are you actually seeing, because of that, synergistic?
We actually don't see any enhancement in reduction in knockdown of any of the genes or any of the endpoints when in combination with standards of care.
All right, well, we're at the slightly past the top of the hour. Is there any, if there's an immediate question, I can ask that, but otherwise I will leave it. Yeah, so I will leave it at here. And Dr. Salzman, thank you again for your excellent and really comprehensive review of the program at Gatehouse. And I want to thank all the attendees for having joined us for this webinar. And I encourage you to attend our next series in pulmonary research. And thank you very much.
Great. Thank you, everyone.